Hi everyone, my name is Mara Burrows. Today is April 11, 2020, and this video is for my Bio 225. This is for week 5, lab 8. We are doing carbohydrate fermentation testing today. So just to start, I have my red phenol broth already. There's 6 of them. I've labeled each one. There is going to be one for the S-epi, one for the S-seri. One's labeled fructose, the other is labeled mannitol, and the other is labeled glucose. So I have 6 in total. I have all my supplies with me. I have my stand, candle, I have my pipette and the Durham testing tube soaked in alcohol. I have my carbohydrate fermentation testing, which I will go ahead and open. So let's get started. Okay, so to start, I have my S-seri vial. Number 1, it's labeled glucose on there. I'm gonna go ahead and open this up and put it over the flame. Carry this. Just put it over the flame. Okay. Next, what I'm gonna do is I'm gonna go ahead and transfer half of the glucose powder into this vial. Okay, so we put about half in. I'm gonna go ahead and pass it through here again. And I'm gonna close it up and shake it around. Next, I'm gonna take the S-epi and do the same. I'm gonna add the glucose to the S-epi. Open it up, over the flame. Add the glucose. I'm gonna do it over the flame. And we are gonna close this up. And again, shake it up. So we are gonna do this for the other 4 vials. Okay, I have went ahead and I have put the glucose in the S-epi and the S-seri. What I'm gonna do next is I'm gonna go ahead and place this Durham vial with the opening side down against the tube and go from there. Okay. So again, tube, we're gonna kind of slide that in slowly. I'm gonna make sure that there are no bubbles, which it doesn't look like there is. And next, I'm gonna go ahead and take this one is S-epi. So I'm gonna take my culture broth for S-epi, which is right over here. I've already had my pipette soaking in alcohol. I am drying it up right now in this towel. In the meantime, while that dries, I'm gonna light my candle. Okay. I've lit my candle. I'm going to take my pipette, which has been dried. I am going to sanitize the top part of this by going over the flame. And I'm going to take some of the broth and do 4 drops. 1, 2, 3, 4. Into here. I'm going to close this. Since this is still sanitized, I'm going to go ahead and grab my other S-Epi and do the same. Okay, so I have all 3 S-Epis done and completed. Now I am going to start on the S-Seri and go from there. As you can see, this is my S-Seri broth. I'm going to go ahead and start the process. We're going to take number 1, which is glucose, and we're going to do the same process. So to start, I'm going to take my Durham tube, side open down, and place that in here. I'm going to make sure that there's no bubbles in the tube. So just move that around to get all the bubbles out. Okay. Next, I'm going to take the S-Seri broth. I'm going to go over the candle with it. Take my pipette, which has been soaked in alcohol and dried out. Dry it up just a little bit. And I'm going to take 4 drops. 1, 2, 3. Okay. I'm going to close this vial. And we're going to go ahead and do the other vials. Okay, so I just finished putting the S-Seri broth on all 6 vials, or all 3 vials. 6 total with the S-Epi and the S-Seri. I do have them right here. I'm going to let this incubate for at least 12 hours. Follow that up and see how it looks. If it is not matured after 12 hours, I will give it another 6 hours, and then we will see the results in the result video. Yeah, so we'll let it incubate. Thank you.